So very briefly, some of the planned exoplanetary missions. You see here the timeline. At the top, you see we are moving from astrophysical characterization, so characterizing the star and getting the basic properties of the planet like mass, radius, and orbit. And then moving forward with time, moving into more detailed characterization of the chemistry of the atmosphere, the climate, clouds, hazes, surface pressure, temperature, etc. The colors at the bottom, we're moving from present day and near future missions like K2 and TESS, Plato's coming soon, and then into future missions like in the near future JWST, and then future giant telescopes like the ELTs or even Hubble and LUVOIR. Sarah Seeger will talk more about the telescopes. This plot comes from a paper by Fuji et al. So I'd like to show you briefly some recent works from the group that I work at in exoplanets and atmospheres at the German Aerospace Center. First of all, you see here, this is a study investigating the potential habitability of a very interesting super-Earth called K2-18b. So the black text at the bottom, this planet is a super-Earth it receives 94% of Earth's incoming irradiance, and it orbits an M2.8 star. So if you look at the figure, so what you see here is wavelength on the x-axis, and what you're seeing on the y-axis is the depth of the transit. So how much does the atmosphere contribute? Yeah, so when you have a transit, the planet moves in front of the star, and the bulk planet will block a large portion of the light, but the atmosphere will also block. Typically, you see here, well, you see the scale in parts per million of the light blocked, and the different colors in the plot you see are different types of atmospheres assumed for this planet. We don't know what atmosphere it has, but we can assume different atmospheres and do a model study. So what you see at the bottom of the plot, we have heavy atmospheres like CO2. So a CO2 atmosphere has a low scale height. It doesn't extend very far over altitude compared to light atmospheres. Light atmospheres extend over large spatial scales. So you see the different atmospheres assumed. The heavier atmospheres are at the bottom, and the medium heavy atmospheres are in the middle, the red line. And what you see at the top is the purple line is an assumed light atmosphere, a hydrogen-helium atmosphere. Light atmospheres extend over large distances and lead to larger features in the transmission spectrum. And you see this here. You see the purple lines, for example, at 1.5 microns, they have much larger spectral features. They go from 2850 up to 3000 ppm, and the other heavy atmospheres, they have much smaller band features. Interestingly, you see the black points here are data points measured by this planet for different telescopes by the study by Bjorn Benneker 2019. And the first thing you see here is that only the purple points, only the purple line fits the black points at 1.5. So this is an indication that the model results are suggesting a light atmosphere, best fitting the data. So they fit very well at 1.5 microns, but if you move out to the right and you look at the two data points on the right-hand side at 3.5 and 4.5, you see especially the 3.5 doesn't fit very well. But in this model study, we did additional model runs with a light atmosphere where we lowered the methane amounts, and then there were some model runs where we could fit all data points. So it's suggesting, at least this study, a light atmosphere with low methane. So this is interesting. It tells you even with the first data we are getting now, we can at least constrain, we can say something maybe about the atmospheric composition. So the second study I'd like to show you also shows a transmission spectrum, but this time it's for the exoplanet LHS 40b. So this is a super-Earth with 41% of Earth's irradiance, and it orbits an M4.5 star. And what you see here, again, we have wavelength on the x-axis and transit depth in parts per million on the y-axis, and you see different model scenarios, the different colored lines. So with varying amounts of methane, so low methane in blue, medium methane in green, and high methane in orange. And the dashed lines show differences in the hazes assumed. And what you see here is that the black dots, which are observations from the Hubble Space Telescope by Edwards et al., and you see that they fit in the region between 1.2 and 1.5 microns. The model studies in orange and purple, and you see that the model studies here are suggesting that the water and methane bands for these atmospheres fit best to the data. Whether we're seeing water or methane is difficult to say, but we can say it's likely one or the other that we're seeing, and we probably need more data or better, higher resolution data to figure out, like, is it water or is it methane that we're seeing? So the conclusions of habitability, it's a central topic in biology, geology, and exoplanetary science. The boundaries of the habitable zone are potentially complex and depend upon climate feedbacks. It's a subject, habitability and biosignatures, which is rapidly expanding in exoplanetary science. Okay. So I'm going to move on now to biosignatures and the diversity of life on Earth. So what you see here are pictures of life on Earth. You see how diverse life is. At the top left, you see sponges, which are very old. On the right, you see ancient life. The trees in the middle left are from California, and the redwoods, they are the oldest living, or the largest, sorry, living organisms. You see very small, like viruses and microbes, bacteria, and people argue, are viruses, can we define them as life or not? At the bottom left, you see a Venus flytrap plant, so plants which behave like animals, so life is very diverse. The challenge of defining life, you see here some of the properties, maybe you learned them at school, of life. So homeostasis, so life regulates its environment. Life has organized cells. Life has a metabolism. It grows, it adapts to stimuli, and it reproduces. Some of the problems of these definitions is that, for example, fire maybe also has a type of metabolism. Yeah, I mean, fire like takes raw materials like wood and burns them and makes energy. It's like a metabolism. And crystals have internal repeating structures, ordered structures. So there are problems with having a unique definition for life. One modern definition of life was discussed by NASA in 1994 in a workshop. They said life is a self-sustaining chemical system capable of Darwinian evolution. So life needs energy from the star or from hydrothermal vents or from radioactive decay. Life needs complexity to build DNA from carbon chains, but other atoms also form chains like silicon. And all life on Earth needs water as a solvent for metabolism, and other solvents have been thought about, like ammonia and methane. Life also needs time to develop. It likes constant conditions. It needs nutrients, the so-called schnops, and it needs some protected conditions from harsh UV or from cosmic rays protection. Here's a summary of life's requirements. So the schnops on the left, energy, appropriate conditions, and a solvent, water. So some examples of biosignatures, life indicating signals. So these are general examples on the left-hand side. So in situ examples, of course, the blue circle. You can look at fossils, for example. You can look at the pattern of carbon numbers. You can look at, so chirality is a measure of symmetry. So my two hands are non-chiral because chiral means I can't superimpose them one on the other. If I try to superimpose my right hand on my left hand, it doesn't fit because they're chiral. Life favors certain isotopic signatures also of carbon. On the remote side, you have like SETI looking for advanced life signals. The more interesting for my work are spectral signatures in the yellow box. So the red edge refers to the increase in reflectivity of vegetation, of plants. Chemical physical disequilibrium refers to, for example, in Earth's atmosphere, there are large amounts of methane, which is a reducer, and also present are large amounts of oxygen, which is an oxidizer. These two molecules are not very consistent with each other. They would destroy each other relatively quickly. But on Earth, in the atmosphere, they are present in large quantities because they're both supplied by life. Methane comes from methanogen bacteria and oxygen comes from photosynthesis. So without life, these two compounds wouldn't be present in such high amounts. So their simultaneous presence is a biosignature. What I'll talk more about is written in red here, the so-called atmospheric biosignatures. So the ideal atmospheric biosignature has no abiotic sources. That means it's only formed by life. It's not formed by non-life. Non-life means abiotic. In the spectra, it has a strong absorption band, which is clear. Yeah, so the band does not overlap with other bands, so it's a single band and you can attribute it to a single species, ideally. And what you do, you have to retrieve the band. You get data, spectral data, and you have to retrieve it and convert that into pressure and temperature and composition, so it should be easy to retrieve. At the bottom here, there's a nice review on atmospheric biosignatures by Eddie Schwieterman in 2018.